In the vast, microscopic theater of life, a silent revolution has been unfolding, one that has fundamentally rewritten the rules of biology. Welcome to the era of CRISPR-Cas9. By 2025, what began as a curious bacterial defense system has transformed into the most powerful tool for precision healing in human history. Its story starts in the microscopic world of bacteria, where an ancient immune system evolved to fight off invading viruses with surgical precision. When a virus attacks, bacteria don't just survive, they remember. They capture fragments of the enemy's DNA and store them in their own genome. These storage sites are known as CRISPR arrays, a living library of past infections that serves as a molecular most wanted list. To execute the defense, the bacterium deploys Cas9, a programmable protein that acts as molecular scissors guided by an RNA map. Once it finds a match, it cuts. By severing the viral DNA, it effectively neutralizes the threat before it can ever replicate. This simple bacterial defense laid the foundation for a technological leap that would eventually earn the Nobel Prize. The road to discovery was paved by decades of collaborative curiosity. Starting with a mystery in an Osaka laboratory in 1987, Japanese scientists first noticed mysterious repeating sequences in E. coli, but their function remained a complete enigma for nearly 20 years. It was Francisco Moica who identified these repeats across different species, finally coining the term CRISPR in 2002. By 2011, Emmanuel Charpentier discovered the final piece of the puzzle: trachRNA, the essential component that makes the system work. In 2012, Doudna and Charpentier published their landmark breakthrough, proving that CRISPR-Cas9 could be programmed to cut any DNA we choose. Their work revolutionized biology almost overnight, culminating in the Nobel Prize in 2020 and a global explosion in genetic research. By 2023, the first CRISPR therapies for sickle cell disease received landmark approvals, proving the technology's life-saving potential. Today, in 2025, that movement is global, setting the stage for the next phase, understanding the incredible mechanism behind the tool. To understand how we edit life, we must look at the technical precision of the CRISPR-Cas9 complex in action. It begins with the guide RNA, a custom-designed map that tells the Cas9 protein exactly where to go within billions of base pairs. Once a match is found, Cas9 binds to the DNA and unzips the double helix, preparing for the most critical step. Cas9 executes a precise double-strand cut, acting as a molecular scalpel that disables the target gene or opens the door for a fix. The cell immediately tries to heal the break. Through a process called NHEJ, it can glue the ends back together, often disabling the gene. But if we provide a repair template, the cell can use it to replace a faulty mutation with a healthy sequence, a process known as HDR. This combination of precise targeting and cellular repair is what makes CRISPR the most versatile tool in the geneticist's toolkit. With this mechanism understood, scientists have turned their focus to some of the most challenging genetic conditions, including cystic fibrosis. Cystic fibrosis is a devastating genetic disease caused by mutations in the CFTR gene, leading to thick, life-threatening mucus. In the most common mutation, the CFTR protein misfolds, failing to transport chloride and causing a buildup of sticky mucus that clogs the lungs. Microscopic view of lung epithelial cells. Mucus-filled airways versus clear, healthy airways after CRISPR correction. Deep blue and teal contrast. The challenge is delivery. Scientists are using engineered AAV vectors and lipid nanoparticles to carry the CRISPR scissors directly into lung stem cells. As the edits take hold, healthy CFTR proteins are produced, chloride transport is restored, and the heavy burden of the disease begins to lift. By 2026, the CF-EDIT clinical trials have shown remarkable success, moving us closer to a permanent cure for those previously left with no options. While challenges in delivery remain, the results are clear. We are witnessing the dawn of a new era in compassionate genetic medicine. But the genetic revolution doesn't stop with human health. In 2025, CRISPR is expanding into entirely new frontiers. In 2025, we are moving beyond traditional gene editing into applications that were once the realm of science fiction. We can now use dead Cas9 to turn genes on or off without ever cutting the DNA, offering reversible control over our genetic expression. In agriculture, CRISPR is creating climate-resilient crops, rice and wheat designed to survive extreme drought and feed a growing world. In oncology, enhanced T cells are being programmed to hunt and destroy cancer with unprecedented accuracy, transforming immunotherapy. From rapid diagnostics to personalized synthetic medicine, the scope of CRISPR's impact is expanding to every corner of our lives. The genetic revolution is no longer a dream of the future. It is the reality of our present, a testament to human ingenuity and hope. The genetic revolution is here. Welcome to the future of healing. Thank you for joining us on this extraordinary journey.